Cardiac arrest occurs when a person's heart stops beating and is fatal if left untreated. While occurring in over 300,000 Americans every year, less than 5% of people actually survive with good neurological function. So suffice to say that cardiac arrest is a major public health issue. And today I'll focus on one form of cardiac arrest called ventricular fibrillation. I believe I have the wrong slide up, but a form of a healthy heart rhythm is one form, and then another form of cardiac arrest is called ventricular fibrillation. And while there are other forms of cardiac arrest, ventricular fibrillation is the only form that relies on receiving an electrical shock to restart the heart to a healthy heart rhythm. And outside the hospital, anyone can deliver a shock using an automated external defibrillator, or you may know it as an AED. So when someone suffers a cardiac arrest, we are taught to perform CPR by delivering shocks at a set time interval. However, this approach of delivering electrical shocks at set time intervals is analogous to the bygone era of using MapQuest for driving directions. MapQuest directions were static. We printed them out and they only gave us one route to our end destination. And in the context of treating someone with ventricular fibrillation, the starting point is grabbing our AED and the end destination is shocking them back to life. Of course, now GPS systems update in real time and give us turn-by-turn directions so that if the conditions change, our GPS will allow us to adapt. And similarly, our AEDs have voice command technology and the ability to analyze data in real time, but yet we have not been able to customize our CPR approach to that individual. And my research seeks to change that. We use a technique that analyzes the height and frequency of the ventricular fibrillation waveform to measure the amplitude spectral area in real time. And that is AMSA. AMSA is our GPS during CPR because it allows us to assess the quality of chest compressions and precisely time the delivery of electrical shocks to that moment in which our shock has the greatest chance of restarting the heart. So in short, if the conditions change during CPR, AMSA allows us to adapt. And in our lab, we performed a study that found that our CPR approach using AMSA outperforms the standard CPR approach. Animals that had the timing of their shocks guided by AMSA had increased survival and improved heart function compared to that standard approach. But most strikingly, that standard approach that had predetermined shocks had 2 to 3 times as many shocks delivered, which meant that those shocks didn't restart the heart and they needlessly damaged the heart tissue. So what we found is that AMSA can bring CPR out of that antiquated MapQuest era and tailor electrical shocks to the individual. And there's currently an ongoing clinical trial using AMSA to guide the timing of electrical shocks. So if our findings from the lab can translate to the clinic, we can see our survival rates increase by 20%, which is astounding. So I am excited for the role of AMSA in CPR. Thank you.
Thank you so much, Salvatore. That was excellent and definitely an excellent example of rolling with the punches and thinking on your feet. Our next speaker is going to be Sarah Grace Dennis Little. Sarah Grace received her Bachelor of Science in Biomedical Engineering from Tulane University and her PhD in Bioengineering from Clemson University. She is currently a postdoctoral scholar at the Medical University of South Carolina and is also pursuing an MBA. Today, she will be telling us about her research improving radiotherapy outcomes for cancer patients. And Sarah Grace, you may begin when you're ready.
You have cancer. 3 words none of us want to hear, but unfortunately, most of us know someone who has. Over half of cancer patients receive radiotherapy treatment. While extremely effective in killing the cancerous tumor, radiotherapy has a cost. It damages nearby healthy tissues. Radiotherapy protocols depend on a delicate balance between effectively diminishing the tumor while also limiting the toxicity to normal healthy tissue. Radiation toxicities affect 50% of treated cancer patients with dangerous side effects such as difficulties swallowing or breathing. Radiotherapy is so effective because it uses a high energy source to kill tumor cells by directly damaging their DNA. In this process, the high energy radiation beam also produces a burst of free radicals, reactive molecules that continue to attack nearby cells. These free radicals, however, do not discriminate between healthy cells and cancer cells. They attack and kill everything in their path. In fact, free radicals account for 2/3 of all radiation damage. However, when these free radicals attack healthy tissues, it results in radiation toxicities. This secondary damage is just accepted as the cost of radiotherapy. There are no current treatments to prevent the damage to healthy tissues surrounding the cancerous tumors. But it doesn't have to be this way. Our lab investigates how we can best protect healthy tissue during radiotherapy. Our goal? Hit the tumor with a radiation without sacrificing the surrounding healthy tissues. We found out this is possible. Our lab developed a novel molecule, hiraloxamer, that physically binds to the cell membranes of healthy tissues and shields them from the damage of hostile free radicals. Hiraloxamer in no way interferes with the ability of the high energy radiation beam to destroy the targeted tumor. We investigated hiraloxamer in small animals, specifically rats. We exposed the animal's right lungs to a single dose of a radiation known to cause lung injury. With no other treatment, the animals became extremely ill and developed lung damage, not just in the targeted right lung, but in the healthy left lung as well. See the bottom left image. In a human, this type of damage causes difficulty breathing, and if left untreated, can become so severe supplemental oxygenation may be required, or even death of the patient can occur. When we gave the animals a dose of hiraloxamer prior to irradiation, we found a significant decrease in the damage to the surrounding healthy tissues. See the bottom right image where the lung damage is localized to the tumor area. The reality is that cancer will continue to be one of the most destructive diseases facing modern mankind. We don't have a cure yet, but we do have a way to improve treatment and save more lives. Thank you.
Thank you so much, Sarah Grace. That was fantastic. Our next speaker is Andrea Morgan. Andrea is from Southern California, where she began her research in the field of addiction. From there, she became interested in the science of pain and trauma. She is now a PhD student in behavioral and systems neuroscience at Oregon Health and Science University, where she is investigating non-somatic influencers like light and scent and the effects that they have on chronic pain disorders. Today, she will discuss how light contributes to chronic migraine pain. And Andrea, you can go ahead when you're ready.
Okay. A lot of people don't like pain, but we feel it anyway. So what's the utility of it? Well, acutely, pain alerts us to tissue damage, as well as the need for medication and rest. Ultimately, it encourages us to protect ourselves. Chronic pain, things like migraine, is different though. It doesn't provide information that's useful, and it isn't necessary for survival. It's just pain for the sake of pain. This is why it's important to understand how chronic pain develops and progresses, but that's not the easiest task. One reason chronic pain can be difficult to study and to treat is because it's not uncommon for people with chronic pain to develop something we call sensory hypersensitivity. What that means is that sensory signals, things like light, for example, somehow become amplified to the point where they're actually able to exacerbate already existing pain. But it's not like these people are staring at the sun and then experiencing pain. This is everyday levels of light like you'd find at home, in an office, or a store. Nothing we have reason to believe would exacerbate pain. For the longest time, doctors and researchers knew this was happening in people with migraines, for example, but it's never been clear why, even though around 80% of people with migraines experience this during migraine attacks. This misunderstood symptom has had doctors thinking that this pain was the result of everything from anxiety to psychosis, but that's starting to change. A couple of years ago, my lab discovered that pain facilitating and pain inhibiting neurons in this part of the brainstem in the middle panel with the purple circle around it act the same in response to light as they do in response to other things that cause pain. So in this area of the brain, the response we have to cutting our finger is the same one that people with migraines might have in response to light. This was huge. What this meant for all those people dealing with this sensitivity to light is that their pain is as real as any other pain. This symptom isn't just the result of anxiety or being a little crazy, but this data came with a ton of questions. I mean, we had this evidence that light can really exacerbate pain, but how? And that's where my work is at now, trying to understand how the brain allows light to get from the eye all the way back to the brainstem to exacerbate pain. And I do this by working with animals that have migraine-like headaches, analyzing pain-related behavior, and by doing immunohistochemistry, which is just a fancy way of saying that I look at where in the brain there's evidence of activity that's specific to the animals with the migraine-like headaches. We now have evidence that brain structures that are involved in other types of pain, like the rostroventral medial medulla, are also involved in this response to light. No Knowing this information will allow us to describe which structures and connections in the brain make possible this heightened sensitivity, and we call these descriptions of structures and connections in the brain neurological circuits. Once we can describe the neurological circuit, we'd have a much better idea of what to target in designing treatments for this kind of pain. Some have said that this type of pain is all in the patient's head. Based on our data, I think my lab would agree, but we'd also add that it's a bit in the brain stem as well. But to be fair, that's true of all pain.
Thank you so much, Andrea. I know we all learned something from that excellent talk. We will next hear from James Quinn. James received his Bachelor of Science in Biology from the University of Sussex and his PhD in Medicine at the University of Manchester. Now he is a Neurology Research Fellow in the Alzheimer's Clinical and Translational Research Unit at Massachusetts General Hospital and Harvard Medical School. Excuse me. Today, James will discuss his research on neuropeptides in Alzheimer's disease and how these proteins may help inform future diagnostics and treatments. James, you can start whenever you're ready.
Thank you. Imagine looking up at all the stars in our galaxy. Really, you are looking back in time, as the light takes 2.5 million years to reach you. Now, our brains contain approximately 100 billion nerve cells, equivalent to the number of stars in our galaxy. And when you count the number of connections between our nerve cells, also known as synapses, multiply that number by 40,000. Big, right? My name is James Quinn, and I crossed the Atlantic to research Alzheimer's disease, the leading cause of dementia worldwide. Much like stars, the nerve cells in our brain hold a window into the past. However, in dementia, our synapses start to break down, resulting in the loss of nerve cells, leading to a gradual decline in memory, issues with daily activities, and loss of independence. Someone who has died from Alzheimer's disease has lost roughly 10% of their brain, equivalent to the weight of an orange. Alzheimer's disease can only be accurately diagnosed after death. So I want to understand why this happens and develop new ways of diagnosing this happening real time in people living with Alzheimer's disease. Improving Alzheimer's diagnosis will increase our chances of identifying new therapeutics like aducanumab to treat Alzheimer's disease. I study neuropeptides, an important signaling molecule in the brain that travels between nerve cells through synapses. We and others have discovered that some neuropeptides are decreased in the brain and cerebrospinal fluid from patients who had Alzheimer's disease. We collect the cerebrospinal fluid, which comes into direct contact with the brain by performing a lumbar puncture, also known as a spinal tap. These neuropeptides are really exciting, as they may allow us to study synaptic health in real time in healthy patients and in people living with Alzheimer's disease. However, they aren't very abundant at all and really represent finding a needle in the haystack. I develop ultrasensitive techniques to detect these neuropeptides in the cerebrospinal fluid. To do this, we use a technique called mass spectrometry. Here, we add the cerebrospinal fluid, heat it up to turn it into a gas, add some ionizing charge using an electron gun, accelerate the molecules through an electric field, and based on their size, they will separate out, which allows us to detect the individual ions. Herein lies the brilliance of mass spectrometry. It breaks apart the haystack to help find the needles. Using mass spectrometry, I've identified that specific neuropeptides may play a role in the progression of Alzheimer's disease. By identifying and tracking neuropeptides important to Alzheimer's disease progression, we are unlocking promising new avenues for diagnostics and therapeutics. My work has the potential to become an important and accurate diagnostic test for diseases that affect the brain and Alzheimer's disease. We also hope to develop new treatments targeting these neuropeptides to help protect synapses, neurons, and keep your brain healthy, with the aim to keep those stars shining to help you make the most of your golden years. Thanks for listening.
Excellent, James. Thank you so much. Our next speaker is Faiza Siddiqui. Faiza completed her bachelor's and master's degrees in biotechnology in Delhi, India. She then completed 2 postdoctoral fellowships, one at Pennsylvania State University and the other at University of South Florida. Currently, she is a research associate in the Department of Internal Medicine at USF. Faiza will share with us today her research on anti-malarial drug resistance. And you may begin when you're ready.
The largest killer of children, that is malaria, kills a child every 30 seconds. Each day, over 3,000 children die due to malaria. 300 to 600 million people are infected with malaria, leading to over 1 million deaths every year. WHO reports that these families in Africa, where most deaths are, they spend a quarter of their annual income on malaria treatment. So it does not only have its toll on the lives lost, but also on the social, economic, and intellectual output of these families and children. In absence of a successful vaccine, we have to rely on anti-malarial chemotherapy. And currently, WHO recommends artemisinin in combination with other partner drugs as the frontline treatment for malaria. You can appreciate the importance of artemisinin by the fact that Professor Yu Tu was awarded a Nobel Prize for her discovery of artemisinin years ago. So artemisinin was for a long time treated as the hero for malaria treatment. But eventually, to our dismay, in around 2008, parasites with artemisinin resistance started emerging in Southeast Asia. What makes these parasites resistant is the question that we are trying to address. We found some novel mutations in a propeller-like protein that show association with artemisinin resistance. So when we introduce these mutations into sensitive strains, they become resistant. And they do so by an upregulated better response towards the DNA or protein damage that is caused by artemisinin. In addition, we also found mutations in a hemoglobin digestion enzyme that show association with artemisinin resistance. That makes sense because hemoglobin digestion is essential for artemisinin activity. So if you block hemoglobin digestion, you eventually block artemisinin activity and the parasites become resistant. So overall, artemisinin resistance is an orchestra that involves multiple partners in crime. And to identify and validate these markers is absolutely crucial to monitor as well as contain the spread of this drug-resistant malaria from Southeast Asia to Africa that can be absolutely catastrophic. This would also help us in identifying better intelligent and better drug policies. Thank you.
Thank you so much, Faiza, and thank you, everyone. We apologize for the technical difficulties that we were having at the top of the flash talks. And we'd ask Sal to come back on camera if you would.
Yeah.
Salvatore, the judges are asking if you would like to redo that just one more time, just so we have a clean cut of this.
Sure. So that slide is actually the previous slide that I sent you. I think there was an updated one with AMSA written on it.
I see.
Yes.
And do you have that potentially on your screen?
No, that's okay. We can go ahead without it. All right, I have to pull up the Zoom again.
Perfect. Then just let us know when you're ready and we will set a timer on our side.
Okay. All right. Cardiac arrest occurs when a person's heart stops beating, and it's fatal if left untreated. And while occurring in over 300,000 Americans every year, less than 5% of people actually survive with good neurological function. So suffice to say that cardiac arrest is a major public health issue. And today I'll focus on one form of cardiac arrest called ventricular fibrillation. On the left side of the screen, you'll see a normal, healthy heart rhythm. And on the right side, you'll see a form of cardiac arrest called ventricular fibrillation. And while there are other forms of cardiac arrest, ventricular fibrillation is the only form that requires an electrical shock to actually restart the heart. And outside the hospital, anyone can deliver an electrical shock using an automated external defibrillator, or you may know it as an AED. And so when someone suffers cardiac arrest, we are taught to perform CPR by starting chest compressions and using that AED to deliver electrical shocks at a set time interval. However, this approach of delivering electrical shocks at a set time interval is analogous to the bygone era of using MapQuest for driving directions. MapQuest directions were static. We printed them out, and they only gave us one route from our starting point to our end destination. And in the context of treating someone with ventricular fibrillation, our starting point is grabbing that AED, and our end destination is actually shocking them back to life. And of course, now GPS systems have updating in real time, and they allow us turn-by-turn directions, so that if the conditions change during driving, the GPS will allow us to adapt. And similarly, our AEDs have voice command technology and the ability to analyze data in real time, but yet we still continue to deliver electrical shocks at a predetermined interval, and we're unable to customize our CPR approach to an individual. So my research seeks to change that. We use a technique that analyzes the height and frequency of that ventricular fibrillation waveform in real time to measure what we call the amplitude spectral area, or AMSA. And AMSA is our GPS during CPR because it allows us to assess the quality of chest compressions and precisely time the delivery of electrical shocks to that moment in which the shock has the greatest chance of restarting the heart. In short, if the conditions change during CPR, AMSA will allow us to adapt. And in our lab, we performed a study that found that our CPR approach using AMSA to guide chest compressions and electrical shocks actually increased survival and improved heart function compared to the standard approach. But most strikingly, in the standard approach, there were 2 to 3 times as many shocks delivered, which meant that those shocks didn't restart the heart and they needlessly damaged the heart tissue. So what we found is that AMSA is able to bring CPR out of the antiquated MapQuest era and tailor electrical shocks to an individual. And there's currently an ongoing clinical trial using AMSA to guide electrical shocks. So if we can take what we found in the lab to the clinical setting, we can improve survival by 20%. So I, for one, am excited for the role of AMSA in CPR. Thank you.
Thank you so much. Thanks to all of our excellent presenters who did a great job in just showing us what science communication is all about and how we can communicate even outside of our fields. I will now ask all of our judges to come on camera. And judges, you're going to ask your questions to each competitor, please.
Jessica, I'm happy to start off. This is Joel. First of all, congratulations to all. Great presentations from everyone. Just a couple of clarification questions. I'll start with Sal. Tell me a little bit about some of the obstacles that you see in advancing this into the clinical stage, or even into the side of the wall stage where you see every other AED. What is the obstacles? The data seem pretty obvious from an AMSA technique versus an older technique. Tell me what you see as obstacles of getting this into clinical practice.
Sure. One of the major obstacles is that ventricular fibrillation is probably accounts for only 20% of cardiac arrest forms. So one of the things is actually making sure that the AED is properly recognizing that a patient is in ventricular fibrillation. So AEDs can do that currently, but that's one of the major components. And then there are other things like having bystanders apply the actual electrical pads to the chest. The placement of those pads somewhat may distort the actual signal, and so that signal is pretty dependent on where they're placed. However, that being said, the AMSA is able to use its own baseline and then determine from there when to shock. So there are certain obstacles regarding the lay people and the general public's use of the AED.
Got it. And just one more follow-up question, if I may. Is the benefit outweigh the risk given the potential for user error on the layperson side?
Yes, I believe so. Because the current trial that's going on in Italy actually accounts for this. So what they have done is that they will have the AMSA going, and then if there are no shocks indicated by the AMSA, then there's actually a time maximum so that they'll deliver a shock. So it's not that the patient would never receive a shock from AMSA, but there would always be a safeguard to ensure that the patient was receiving adequate shocks.
Great, thank you.
Yep.
All right, thank you. Are there other questions from the judges?
Yes. Can you hear me okay? My question is for Faiza. I'm interested in your understanding of the mechanism for the artemisinin resistance. Do you think that that will ultimately lead to some sort of secondary therapy that you'll combine with the original one, or some entirely new class of drugs ultimately, if you can identify the mechanism?
That's a very important question, actually. So identifying the mechanism, exact mechanism, is essential to, safeguard the parasites from developing resistance. So artemisinin, actually, it's a very, I can talk on for hours about artemisinin mode of action, but it is such a fascinating drug because it targets not just one or two, it targets multiple pathways in the parasite. And that is the reason that even the resistance, has not evolved completely, because it's not just one protein. If you mutated, it becomes completely resistant. And that is, I think, in a way, good news. And that is the reason why full-blown resistance has not developed. But as far as for now, what we know of artemisinin mode of action is it involves proteasomal machinery of action. It involves oxidative stress of the parasites. It involves vesicular transport that reduces hemoglobin ingestion and digestion. And it also involves DNA damage. So there are so the silver lining is that, so there are a recent study there was in Lancet where they reported triple combination. So far, we are only using in field double combinations, as you mentioned. So that is only using artemisinin with just one partner drug. And if for example, for other previous drugs, there has already been resistance in that area, but if it has been maybe a decade or two that has passed, we can reshuffle those drugs. So all these strategies are taking in place, and the data that we have from the triple combination therapies is looking quite promising. So that's something that we are all looking forward to.
Thank you.
Okay, and judges, I'll let you continue asking questions of our finalists.
Right, I have a question for Dr. Quinn. What do you see as the biggest obstacles or X factors as you're conducting your research?
I'm sorry, I heard obstacles. What was the following phrase?
Or X factors, or anything that you're concerned about as you're doing this?
Yeah, so there's quite a lot of different things that we're thinking. I think the most important thing for us is that we get cerebrospinal fluid from patients that then we can then get their brains from in the future. So I think that's one thing that we would love, and that is actually something that is not that common. Is what we notice. It's very hard to get cerebrospinal fluid from a person 10 years before they then subsequently passed away from Alzheimer's disease. I think that's probably the number one issue. And then also we deal a lot with pre-analytical factors. So basically this is anything that is done to the cerebrospinal fluid sample before we get to measure it, right? So how it's frozen, how it's been put in a centrifuge, all of these different things. If someone has left it on the bench for an hour before they then put it in the freezer, all of those things can have an impact on our results. So it gets difficult when you're looking at samples that are 15 years old, 10 years old. So I think definitely there's a massive concerted effort in the Alzheimer's field to really standardize these processes. So have kind of one procedure that every single lab tries to follow, and then these are as much as standardized as possible. And I think finally the biggest obstacle is really just education. Getting people educated about Alzheimer's disease is super important. And it still blows my mind that a lot of people still don't think dementia is a disease that we can hopefully treat in the future, and hopefully treat with aducanumab and other drugs that are coming in the pipeline. So it's a really exciting time to be in Alzheimer's research. But yeah, lots of kind of X factors, as you say, that are coming in the line for the future.
James, can I ask one additional question too? This is Joel again. Thank you for that answer, and remind me again, as you know, there are several stages of Alzheimer's disease, both the cognition impairment all the way to full-blown dementia and AD. Where are you focused?
So in this study that we're hoping to get published sometime this year, we looked at healthy controls. We looked at people who have mild cognitive impairment. So this is Not everyone who has mild cognitive impairment subsequently goes on to develop Alzheimer's disease. This is showing that they have some issues with their cognition. Cognition is basically just the word we use for everyday brain functioning. So they have a mild impairment there. We're also looking at people who have Alzheimer's disease. And then we are also, and we could define Alzheimer's disease by looking at amyloid and tau, which are two proteins that are known to accumulate in Alzheimer's disease in the brain. And we can also detect them in the cerebrospinal fluid and in the blood. So we characterize them according to that. The mild cognitive impairment is only characterized by cognitive scores. And then the final group of subjects we're looking at is this resilient group, which I find to be the most fascinating. So these are people who have amyloid and tau in their brain, or amyloid and tau in the cerebrospinal fluid or blood, sorry. But they do not have any issues with cognition. So to me, this represents a really, really exciting group to study, because why do they have these proteins that we know accumulate and potentially cause the disease? Why are they accumulating and then not causing any impact on cognition? And we're already seeing some really exciting changes, and I look forward to being able to share the results in the coming year.
Great, thank you.
I have a question for Andrea. Well, first, thank you for your presentation. It was really enlightening, and I don't mean that facetiously, but it was really enlightening. I think so. I'm really interested in knowing more about kind of what you have discovered around the neurological pathways. Do you foresee there being separate neurological pathways that correspond to different sensitivities and sensory function, or like what else have you discovered just from your research?
Yeah, so there's a lot of different pathways playing into pain, because we have a descending pain modulation system. And so all these things from like memory to fear to even depression and things like that are kind of playing into this. When I'm looking at the sensory systems, I was the particular structures, I guess, when it comes to photosensitivity that I was really interested in were intrinsically photosensitive retinal ganglion cells. They send projections from the retina to the amygdala. And the amygdala is kind of this sort of integration point for all the sensory information coming in. And then the amygdala is able to send projections down to the RVM, or the rostroventral medial medulla, which is really where descending pain modulation is kind of like the big focus of it, is that's where it's going on. And so I've been particularly interested in the retina and trying to get more into vision and understanding like how does this information go from one place to another. But along with trying to understand vision better, I'm also trying to understand the influence of like social hierarchies and things like that. And so I have my animals housed tuber cage, and so I've been able to see this. I have what I'm calling a within cage data split. And so I'll have like one male that is like responding like to everything, like super sensitive, and then another male in the same cage that's not responding to anything, just refuses to move, refuses to acknowledge that anything else is going on. And so I was wondering like what would cause that? And so as I was looking through the literature, I realized that dominance actually has a major role in pain-related behavior, and that when you're testing males in front of other males, their behavior, whether they're dominant or submissive, can really influence the ultimate behavior. And so my pain treatment in my males is completely washed out, and we think that it's because of this dominance and this within cage data split, but it makes it look like there's no significant differences in males. So I'm trying to understand like where some of that social influence is coming from, and like where is that getting into the circuit?
That's so interesting, and I can't help but think about the history of eugenics and just even the ways that health inequities continue to show up, mostly between Black and brown patients versus white patients in pain and perception of pain. So very interesting. Thank you for sharing that.
Yeah, and I'll just say real quick, because I think I've been fascinated by the same thing because I came from a psychology department. And that was one of the things we talked about is there was a study with Hispanic males, and it showed like if you ask them a question in this particular way, like machismo kind of takes over and they don't want to answer. And so we're getting really weird statistics about prevalence in Hispanic males in particular, and they think that it's a dominance component. And so we see it in humans as well, to a point where it's actually affecting prevalence rates.
Thank you.
Joel, again, I have a question for Sarah Grace, if I may. Sarah Grace, I'd like to ask you a little bit about sort of the clinical application of your model. You said, I believe there is clinical trials underway right now. Is there any delivery toxicity to your molecule or anything that you're testing now that would either inhibit or throw a blockade in front of any clinical development?
So we are actually not in our clinical trials yet. We are actually preparing for our pre-IND submission next month with the FDA. But the unique thing about this molecule actually is that it has failed a previous clinical trial. And so we actually understand a lot of its pharmacokinetics and all of the adverse events, like the phase 1 safety trials already been completed. And so what the side effects are, you have to keep administration on for over 36 hours, and it gets cleared through the kidneys. So patients must have functioning kidneys in order to clear the molecule. But, we're talking about dosing these patients, a loading dose about an hour prior to radiation therapy, followed by a loading dose that gets cleared in about 4.5 hours. So we're not anticipating any real adverse events from the dosing, the route of administration, or the drug itself. I think the big fear of starting the clinical trial is in the efficacy and making sure that our dose that we tested in the animals translates as the therapeutic dose for these cancer patients.
Got it. Thank you. And so you said 36-hour post radiation.
You have to be on a continuous IV of the molecule for 36 hours, in which case it will actually start, instead of stabilizing your membranes, it will start to permeabilize them and destabilize them, resulting in organ swelling. However, when they did remove the IVs after that amount of time, the organs returned to normal after a few hours. So it was not like a permanent adverse event, just a learning curve on how to dose.
Great. And then what were the failing aspects of the earlier trials that you're avoiding now?
So they actually didn't understand the mechanism of action of the compound itself. So they actually targeted sickle cell patients because they thought that it would break up the blood clots. So it has shown to reduce blood viscosity in vitro. So they kind of ran with that. They, to be honest, did not perform the appropriate due diligence in the preclinical space on it. And in their phase 2 efficacy trial, they did not have a statistical significance of success.
Got it. And then one last question, sorry to hog all your time, but how did you discover the sort of free radical collection aspects of this molecule?
So our lab does a lot of biomaterials research. We're actually part of a statewide grant. So biomaterials has been my realm for years. So we started investigating it in vitro, and one of my screening assays is actually a hydrogen peroxide assay. So we got some promising data on this molecule in that assay where we saw a significant number of cells survived. And we started thinking, well, where in medicine is there a big oxidative stress event? And we got led into the radiation oncology route, and then we decided to go forward, and we did some in vitro stuff, and then we went ahead on the animal studies after that, and now we're running.
Great. Good luck.
Thank you.
All right, thanks so much. Great answers to some great questions. Do our judges have any other questions at this time? Okay, then our judges, I'll ask you to go to your breakout rooms, which will be in your calendar invite. And audience, it is now your time. If you have any questions For anyone here, please either put that in the chat or raise your hand and we will answer those as best we can. Okay, let me see. A question for Faiza. What role does monitoring play in preventing the spread of drug-resistant parasites? And do you think changes need to be made to any existing surveillance techniques that we have now?
Yeah, so monitoring is very important because, so resistance, developing of resistance takes a long time. Like it's not a matter of months, it's like a course of years or sometimes, even decades that the resistance actually evolves. So it is very important to monitor this resistance like in the very beginning. If you see like, for example, some proportion of the parasites start showing resistant alleles, then in that particular area, for example, like in those regions, you can probably, do an alternate drug policies so that, like a drug has been used previously, like decades before, and these parasites have not been exposed to that drug for a long time. So maybe now you can use it in combination with other drugs, so that's very important. So you can catch that in the beginning. And as far as like surveillance strategies are concerned, so we have like field sites in Myanmar, and like there are a lot of field sites in Thailand, and so it's like doing PCRs and doing like Sanger sequencing is not always feasible for in those remote areas. So I think, there is always a scope for, actually there is a company like who is like, has been developing, like, I think for last 10 years, they've been working on this technique. Like you can, instead of doing like Sanger sequencing, if you're able to identify the molecular markers, like right on, like doing an incubation in a tube and just running the DNA, or maybe you don't have to, like, if you can avoid Sanger sequencing and you can make it more cost effective so that, and these are the regions where there is more drug abuse, and that is the reason the resistance develops more. So, all these things are very important. And even on like, so we are also like are in communication with like national policymakers for in Thailand especially, and in Myanmar also. So it is important to discuss with them. So what is the proportion of the parasites right now which are showing resistance, and the resistance is towards which drug? So you should avoid that drug so the resistance is not like highly developed to that. And again, as I mentioned in my talk, like, so it has always happened for all previous drugs that resistance first developed in Southeast Asia and transferred to Africa, where most deaths occur. So if you are, so here also it's developing in Southeast Asia, if you are able to contain it there by constant monitoring, it will really help in, preventing the spread worldwide. I hope that answers the question.
Yes, thank you so much. And we actually have another question for you. If you can comment on the impact of climate change on mosquito vector-borne diseases and whether there's a connection with resistance.
Oh, of course, because like even malaria, right? As you all might be aware that it is an insect-borne like disease. So it is very important, like, and so I would say if not equal, I think like 40-60, so 40% of efforts are actually targeted towards elimination of mosquito, or if somehow we are able to modify them so that like they become like resistant to carry the parasite. So that's how you can prevent the spread of the disease. So of course, like, so since this is governed by climate change and those factors, so this actually also plays in the population structure, population diversity of the mosquitoes which carry malaria and spread malaria. So that also plays like a huge role. So like I remember like I attended a talk where they talked about like, just a matter of like a few kilometers, because of the drastic change of the weathers in some places. So the populations are like, so it is only the female mosquito population that carries the malaria parasite. And they showed that in that particular weather environment, the female population is not much supported. So, so that way is like, it can be modified at that scale also. And as I said, like equal attention has been put on controlling the mosquito population, and you might all be aware of the DDT story, and as well as like what the kind of work that we are doing, like molecular diagnosis. And so both the things are important, and definitely climate change is like, I think it's affecting everything, and malaria is not lagging behind.
Great, thank you so much. And for Andrea, with respect to male dominance, has this been observed in other studies?
Yeah, this has been observed, and it's the Mogul lab at McGill University in particular has reported on a number of different social conditions like that. It's really an interesting area of literature that I had no idea existed, but dominance is turning out to be a very important thing, particularly in males in studies of pain.
Great, thank you so much. And let's see. For Sarah Grace, can you explain a little bit more about the mechanism by which Herloxamer is protecting healthy cells?
Sure. So Herloxamer is a tri-block copolymer, and it's got a hydrophobic core flanked by two hydrophilic tails. And the balance of that hydrophobicity and the hydrophilicity is what determines whether it stabilizes cell membranes or destabilizes and permeabilizes, disrupting cell membranes and causing leaking of the intracellular components of the cell and things like that. So it's this really delicate balance. And so that hydrophobic core of the Herloxamer wants to actually bind to the hydrophobic interior leaflet of the cell membrane. But because of the hydrophilic tails, it's unable to actually penetrate into that leaflet and actually just sits on the exterior of the cell with its hydrophilic tails kind of like this, available to quench incoming reactive oxygen species, free radicals, all of those things. And at the same time, those hydrophilic tails, there's multiple of these molecules on the cell membrane, and they actually interact with each other as well, exerting a slight compression on the membrane, holding it together. And we're actually investigating this in silico now. So we've started a whole molecular dynamics simulation project around simulating these interactions with the cell membrane to try to understand how changing this hydrophobic philic balance of the molecule can exert different interactions with the cell membrane.
Great, thank you so much. Once again, for the audience, you can put questions into either the Q&A or the chat, or you can just raise your hand and we will be able to unmute you. So another question that we have is for James. And this is about what population you think would benefit the most from earlier Alzheimer's screening. So is this something that we would want for just any elderly population to receive, or people maybe who have certain risk factors?
I think that's a really fantastic question. And I know it's a kind of a big debate in the research field, but I think the most important thing to take home is having a diagnosis means, you know, you can plan things, right? And I think that's the most helpful thing. And I know my grandmother, she spent years trying to get her dementia diagnosis, and it's the same story that you hear over and over again. And I think the one thing that excites me most about Alzheimer's disease research right now is, you know, the amount of work that's going into diagnosis. And I really think in 5-10 years, we're going to have a blood or a cerebrospinal fluid test for Alzheimer's disease. I know there's a lot more work to be done in other forms of dementia that aren't studied as much as Alzheimer's disease, such as frontotemporal dementia, vascular dementia, etc. But in terms of the risk population, I think it's really good for anybody, but also specifically if people have some kind of risk factor that they're worried about. I don't think, I'm not a proponent here of kind of population-wide screening, but I think it'll be really important for anyone who's worried about their brain health to go in and be able to get a test and just have that discussion with their doctor and kind of really plan that forward.
Excellent. Thank you so much. A question for Sal. How often are defibrillators now being used unnecessarily, and would your new type of equipment guard against situations where defibrillation is not necessary at all?
Sure. So defibrillators are used for out-of-hospital cardiac arrest, and that's primarily where we see them used. So you find them in the gym, or like schools, or any kind of public building normally will have an AED in it. And the primary goal of using the AED is to, I mean, if anyone's been trained in CPR recently, you'll know that when you pull out an AED, open it up, it starts to actually give you all the voice commands. So it'll tell you, put the pads on the patient and then start doing chest compressions. And what it'll do is actually read the heart rhythm. And so if the patient is not in cardiac arrest, the AED will be able to recognize that, and they'll be prompted to call 911, certainly if the patient's down, but they'll also be able to assess whether or not the patient's in cardiac arrest and then prompt you to do CPR. So the AEDs are being used out of hospital, but it's really the timing of the shocks that is where they're not being used properly, or not being optimized in their use, in my opinion, and as the research has been showing. So the interesting thing about what I'm doing is that these algorithms are already in the AEDs, and different companies have different proprietary algorithms, but all this technology is currently available. It's just not cleared FDA standards because the appropriate clinical trials haven't been used and all that. So this is very exciting research because it's very close to being clinically applicable. The ongoing trial, if there are positive results, we could be seeing this technology being used in a few short years. So it's very exciting to see that AEDs could really start to hopefully improve outcomes from cardiac arrest outside the hospital.
Great. Thank you so much. Those presentations were absolutely fantastic, and great Q&A question from everybody. And now the moment we have all been waiting for. Judges, please come back on screen to announce the Flash Talk winners.
Sorry, I think we lost one of our judges due to his commitment on another panel, but I'll kick this off. Jessica, if you're comfortable with that.
Absolutely, go ahead.
Okay, well, great. First of all, I want to congratulate all 5 of the finalists. All of you did such a wonderful job, and I don't know if the entire audience knows, but this was a group of young scientists who competed in a prior competition in July, just a few months ago, and have now moved on to the big competition here at the Research Forum. So I want to congratulate for all of these 5 for making it into this round, but also for the improved performance from what we saw in July. Really, really wonderful work. Not only did you consolidate your scientific message, your communication style, but also we threw in a little bit of a curve, giving you some questions where you really had to think on your feet. Really outstanding work by all of you. We were tasked with giving some rankings to all of the contestants and contenders, and we weren't able to give 5 first place awards. So we were going down that road, but I do say that we do have some clear winners, and I would just like to say that in our eyes, and I'll let our judges make a few other comments as well, that all 5 of you are incredible winners, and we hope that your research continues on and eliminates some of those obstacles we talked about during the Q&A session. So without further delay, I'd like to announce our tied 3rd place winners. So we had to do this. We were debating about who is in 3rd and 4th place, and also 5th place for that matter. It's really hard to have someone bring up the tail end because all of you are so good. But I want to say that tied for 3rd place are Sal and Andrea. Sal, as you recall, had the VFib AMSA presentation, and Andrea had the light migraine pain presentation. Really fantastic work. And Faiza, your work is also phenomenal, and really you're so knowledgeable about the anti-malarial field that we sincerely hope that your research continues and you are able to continue in that field. But tied for third place are Sal and Andrea. Wonderful job, guys. Second place and runner-up is Sarah Grace with her work in radiation recovery and cell preservation. Wonderful job. I think our entire panel feels that this has strong, very strong clinical application, and we wish you the best. I know from our conversations that your research continues, your clinical work is moving forward in the clinical trial perspective, and you're also trying to move this into the business world as well. So congratulations on second place, but also congratulations on your research, which leaves our number one winner here, James, with your neuropeptides work in Alzheimer's disease. Really fantastic. I think your presentation improved from July. The use of an orange as the size of my functional brain was a fantastic addition. And I just want to say congratulations. Really strong application to the science in the clinical world, and also strong application to what it means for patients. So congratulations, James, for winning the Flash Socks competition. But I also want to say thanks and congratulations to all 5 of you. Just absolutely wonderful job. That's all I have to say. I'll turn it over to Lauren and Dana for their comments as well.
Well, I'll just echo what you've already said, Joel. Thank you so much to all of our presenters. Excellent job. We definitely could see the additional work you put in over the summer. This is such an important skill. I really just want to commend Research America for highlighting this and commend you all for stepping up to this challenge. You will be better scientists and better communicators because of it. So congratulations to you, and please keep up the good work. We need your brains, and we need your accomplishments and insights into the future.
Thanks, Lauren. And I'll add, this was a very, very difficult decision. All 5 of you were fantastic. And for those of us who were able to participate and listen to many of these presentations earlier in the summer, to see how so many of you had also done the work to make your presentations even better when we thought they were so good to begin with, really just goes to your dedication and your talent. And I, for one, am just so excited to see what you're all going to be able to do in the careers they have ahead of you. It's very important to be able to talk about your research in a way that people understand, especially some of these very complicated things. So I really commend you for all of your work. And I just want to say I was personally touched by a lot of these presentations. I thought that you all did a fantastic job in condensing these very difficult to comprehend matters into something that in 3 minutes you can convey to someone, and also give them hope in everything from malaria to Alzheimer's to cancer, that there is going to be hope and new treatments and therapies coming down the line. So I want to thank you again for that. And it's been an absolute pleasure. Thank you.